and we'll get started. So welcome everybody for our last session of RTTI, at least the fall virtual sessions. Hopefully I will see you guys in the spring as well. I'll talk more about that in a couple minutes. But thank you to everybody who sent nice messages already talking about how much you've enjoyed the sessions. So if you guys have other messages you like to pass along, our board loves to hear that. Legislators love to hear that. I love to hear it. And we love quotes that we can send to DOE in our end of year report. So please email your feedback, kind words. And if you like it, please also contact your legislator. So if you do not know who your representatives are, here's a website where you can do that. I pulled it right off of the Georgia government website. We'll admit the rest of these folks. But please contact your legislator and let them know that you have been enjoying RTTI. We are a line item in the state budget, so we would love to be funded again next year. And so if you guys can tell them that this is essential and you're enjoying it, they'll know from every corner of the state. You guys probably, a lot of you have different legislators, so we can get some buzz when they go back in session. And I will post the links that I put in the chat in the Google Drive. So hopefully everybody found those links from last week. Let's see. So I emailed out the equipment survey last week, and a lot of you already returned that. So thank you. That's very helpful for us to see kind of what items we should have for loan, and then what items we should be soliciting for. So if there's anything that's not on that list that you need for your classroom, just send me an email with a list of items. And so as donations kind of roll in, I'll keep an eye out for those things. And I can report back to you if they arrive. Letting some more people in. And there, Jeff has co-host access. Let's see. Okay. Does anybody have any questions right now about the equipment survey or anything?
Is that for when we try to do the labs with our classes later?
So to help you prepare so that you can do labs, biotechnology labs with your classes, yeah. And a lot of that equipment may not be in your classroom. Somebody might have it stored away in a closet. So this is a great opportunity to say, I need to go on a scavenger hunt in your classroom and look through your closet and see what you have. Because I know some people are weird hoarders about things. So
Okay, thank you.
you have permission from me to go raid their closet. Awesome. And yeah, thank you, Erica, in the chat. Anybody, as usual, if you have multiple people watching on the same screen, just please pop in the chat who is sitting in your room together. Awesome. Okay. So I wanted to show you guys a gel electrophoresis simulation. So again, on LabXchange. You don't have to do this right now, but I wanted to make sure that we kind of highlighted it as a resource. So I will pull it up and you guys can kind of see. But this is a good thing to go through with your students, just like the pipetting activity. Cool, and it always gets rid of my student or y'all's view. Now I can't see anybody. That's fun. I know you're there. I hear you. Ah, there you are. Okay. So this simulation for gel electrophoresis, it goes through kind of like the pipetting one. So we click and then it gives you the options for level 1, 2, or 3. So if you have students that say, I've done this already, I'm an expert, you can go ahead and have them try level 3 and see if they really are an expert. But we can look at level 1. So there's the context here. So what do you need to know before the lab starts? And Mark and Trisha will talk a little bit more about this when we get to loading our gels. But gel electrophoresis, it's a separation technique. So you can separate things like DNA and RNA or protein in the agarose gel. And so the agarose gel it's kind of like a sponge on a molecular level. It has little holes. And so the DNA can travel through. And so larger molecules are going to travel more slowly than smaller molecules. And that's according to charge. So you guys will notice you have batteries, and we will be chaining those together, positive to negative, and creating a current. So if you're looking to use this with physical science classes, you can really highlight the voltage and current and all that good stuff. So you'll be loading it at the negative electrode, and then it'll travel toward the positive electrode. So there's this nice review for your students about how it works. And then all the different reagents, pipettes, power supply, all the electrophoresis equipment, which you guys are doing the DIY version today, instead of having the big fancy boxes. But you'll be able to see that it works as well. And then we have the other equipment. So there's an area to make predictions about how you think things will travel. And then you can see on the right, it goes into the protocol. So we talked about how following standard operating procedures and protocols is extremely important. So they can exhibit that for you here. And then we have, the protocol, and they can go back and then go to results and reflection, and then there's a summary. So again, a good resource to assign your students before you do gel electrophoresis. And then if you choose to do the DIY activity with them, you could highlight how the DIY version meets all of the same kind of components of what you see in the simulation. So pretty cool. And some people have joined. Gonna let them in the waiting room. So you guys can go back and complete that on your own time. Just another good resource for y'all. So let's see. Yeah, some Zoom crashing. So today the recording will be all in one block. So Mark and Trisha are sitting there at home. They went to the STEM STEAM conference. So any of y'all that ended up going may have seen them around. I was there Monday presenting for a session. So I saw some of you guys there. So that was exciting to see some people in person for once instead of through the Zoom. All right. So let's see. As far as the spring goes, so the in-person sessions will be at Central Georgia Technical College in Macon on January 26th and 27th. And I'll send you guys some more information on that soon with an RSVP and a calendar hold and some suggested hotels. Probably by the end of the week. I think I put it on Friday to do that for you guys. So, but any questions that you guys have about that, feel free to reach out. So now, Jeff, are you available to speak? I wanted to give Jeff a couple minutes to talk about his new role. So Jeff, before this year, was at Athens Technical College as the Director of Bioscience, and now he is at the Director of the Bioexpression and Fermentation Facility at UGA. So he'll tell you guys a little bit about that.
Okay. Yeah, thanks for the opportunity. I was program chair for bioscience at Athens Technical College for about 18 years, and we had a good outreach program with high school science teachers for several years, but then it kind of went away as a grant went away, unfortunately. But then Megan and Kristen were able to pick up that ball and keep running with it. So the technical colleges, there are 4 main bioscience programs in the state. Ours at Athens Tech was the first, but also Gwinnett Tech and Central Georgia Technical College and Atlanta Tech all have bioscience programs, and they'd love to have your students go there. It's a 2-year technical program that trains people to be an entry-level technician for either research laboratories or a biomanufacturing plant. But now I'm at the University of Georgia, and I'm director of the bioexpression and fermentation facility. And we're a contract facility for usually faculty members at UGA who need products for their research, like a certain protein that they do research on, or cells that they need grown. We do that kind of thing. So we have 4 main procedures that we do: growing cells, either bacteria or yeast or mammalian cells. We also do protein purification. We make monoclonal antibodies, and those are the main things we do here. And if you ever come here, I can give a tour to you and your students, and hope you can visit us sometime.
Thanks,
I was
Jeff.
just up there Friday. I should have, had I known.
At least come by to say hello.
Well, I mean, I took my son to a tour of the College of Engineering.
Oh, okay. Yeah.
They didn't really stress the biology labs in the College of Engineering much, which was disappointing. But
Right.
I don't think that the tour guides knew what to do with those 2 rooms.
So again, this is Terry Bennett, sequence from Glassman Housing for 11.
So yeah, any questions about UGA, the bioexpression facility?
Thank you so much.
Yeah, or the technical colleges too. Technical program. Jeff still knows that very well.
Yes, and they would love to have your students go into their programs. And the jobs are so plentiful now. I mean, always in biotech, your students will have a job if they take that route. There's such a demand for technicians and workers in these biomanufacturing facilities. The job opportunities are really vast.
Any idea if, like, I know Southern Crescent Technical is like really growing, but is there any have you heard of any biosciences coming to that technical program, or possibly?
Is that in South Georgia?
That's in it's Henry County, Griffin,
Okay.
a little south there.
I haven't heard of any, but that doesn't mean
Yeah, because they've been expanding their campuses. I mean, it's in between what would be the Atlanta Tech and the Central Georgia. So it's kind of
right. That would be a good place to have one.
That middle spot there. So
right,
I've been trying to talk to my students about it, and that would be the spot.
That would be good. Takeda is one of the largest biotech companies in the state, and they're not far from there, and they're constantly looking for people. I mean, they have hundreds of.
Are they in Henry County?
They're in.
They have a LabCorp.
I think Newton County.
They're in
Social Circle, Georgia.
Okay, because I don't know if I heard of some division of them. We've got a lot of warehouses, and we have a LabCorp not far from my house, a big place as well. So.
Bioscience programs in South Georgia.
South Georgia, not that I know of, but there again, there's a company down there making biodegradable plastics, and they have a really big demand for workers. So I wouldn't be surprised if one comes along soon. Yes, someone, she wrote it. Yes, Megan wrote that. Danimer Scientific in Bainbridge.
Yeah, and I think one of you guys posted about how Danimer came and spoke at the middle school. So if you guys are down in that area, yeah, you can reach out, and they'll be happy to send somebody to come to the school and talk and maybe do a little lab activity. So yeah, awesome. All right. And did you admit Dave?
No, I haven't.
Okay. I think his computer dropped. Oh, there he goes. Okay. All right. Well, let's see. I'll give Dave a second to get booted up. But we're going to have 2 speakers today. So our first speaker here until 5 is Dave Penicky. He should be loading. And then once you guys get your gels running, we'll hear from Lori Kreimeier from Boehringer Ingelheim. Let's see, Dave, are you here? Still getting loaded.
Can anyone hear me?
Yes, there you
Yes.
are.
Okay, great. All right. Am I still there?
You are still here.
Perfect.
And I will pull up your PowerPoint.
Great. Well, hey, I apologize. I ended up having my schedule got a little mixed up today, so I had to drive. So I pulled over. I'm really excited to have a chance to speak with you guys today. So you guys are doing electrophoresis today?
Yes.
Okay, that's pretty neat. So I was speaking with Megan when I did electrophoresis the first time. I think the technology was a little bit harder to come by, but it's pretty neat that you guys are doing that. And then Megan, am I okay to get rolling, or are there people that you want to wait on?
Yes, you are.
Okay,
Yeah.
great. Well, really exciting to have the chance to speak with you guys. My name is Dave Pinnock. I'm the CEO of Sail Therapeutics. We're based we have two offices, one in Dublin, Ireland, and one in Roswell, Georgia. And, had the opportunity to kind of see if there were some things that we could talk about here that would help share, what some of the work that you guys are doing, trying to, help teach people in this state that have an interest in life sciences, and, what ways might be most helpful from a company perspective, or from a professional perspective, or from a translational skill perspective. So that's a little bit what I tried to focus on today, and tried to make it, a little more practical and hands-on. I'm happy to answer any questions or talk about anything related in this presentation, but that was sort of the focus. So if you have questions, you can feel free to interrupt me throughout or save them to the end. I'm happy to do either way. So next slide. All right. So who am I? Well, and how did I end up in Georgia? I was born in rural Montana and grew up in central Iowa. And so it ended up from there gaining an interest in science and engineering. I ended up going to college in Chicago. I took a job in the tech industry. I worked at a manufacturing facility that made computer chips at Intel. Really hated it. I liked the sort of process of making things, but I just couldn't relate to the products. And, went back to grad school and then switched into biotech, where I really resonate with both the science, how you make these things, and also kind of the outcome of the product. I like, medicines more than I like phones. And so from there, kind of developed my career in the San Francisco Bay Area, and then moved to Atlanta to become the CEO of Sail Therapeutics after I had, a decent amount of experience. And here it's been really cool. We've invented some medicines. We've acquired some medicines that we turned around and sold. And now we're a clinical stage company, and we're studying a rare disease. We're studying brain cancer, osteoarthritis, knee pain, and spasticity. And then, just from a standpoint of kind of how I think about things, I've definitely done the, commercial side and sales and marketing side of biotech, but I really love, the engineering. I really love solving problems. And I think that's, the skill sets that support problem solving can really be put to work anywhere in this industry, which I hope to share with you. I love college football. I was a punter and a kicker in college. So, definitely like living down in the Southeast. People here appreciate college football a lot more than they do on the West Coast. It's been my experience. So next slide. Okay, so this is my family. I'm the little guy on the left in the old picture there, and that's my mom, my brother, and my dad. The center picture is I have wife Adrienne and 3 children who are 7, 10, and 13 years old. That's a picture of us just hiking up in North Carolina on one of the long school weekends. And then the upper right is my parents, my brother and his wife and daughter, at my parents' 50th anniversary. So it's been kind of fun getting to know these kids as they grow up, and I certainly appreciate everything that's done to support teaching them what they learn in school. I think the the access to technology these days and the changes that are happening are incredible. So it's exciting for me to be able to speak with you and hopefully provide any insight that might be useful to students like my children going forward. Next slide. Okay, so Sail Therapeutics, we're a relatively small company now. We have about 25 people split between here and Ireland. When we after we acquired 5 products and we had 5 products on the market, we had about 100 people. We had an office in the Middle East as well. And what I would say is I think people think of the large companies as what pharma is. And what pharma is actually like those large companies kind of gobble up all the small companies eventually. But the small companies are, they're scrappy, they're little, we manage our budgets and, this is the team I work with. We sit in a really small room when we get together. We don't spend a lot of money on facilities, but we do have a lot of fun and we try and solve problems. And then I think what you see is a lot of the small biotechs end up getting gobbled up. I think I read something the other day that 87% of new medicines that have been approved over the last 25 years were actually invented outside of big pharma and then were acquired into big pharma. And so what's exciting for me about being in Atlanta, in Georgia, and having the chance to speak with folks like all of you is that, what it really takes is like these bands of like small people, 12, 24 people that come up with a lot of these innovative ideas. And, it doesn't sort of take a Nobel laureate or a giant university. I think what you see are smart kids that come out of these at a high school, have an interest in science, and then find their way into these organizations and then, invent ways to sort of make medicines better or get something to market. And so it's fun, it's challenging, it's exciting. And I think it leans on skill sets that you guys are already helping people develop. On the right, that's my wife and my middle son, Brandon. We were trying to test a new flavoring. And so, the big companies, they go out and they run all these flavoring studies, and we just sort of gave the stuff to friends, the placebos, just to like see how it tasted. And, that eventually led to a medicine we got approved. We chose strawberry, which was Livispa, and it was launched by another company last year. So, I think for people who are interested in science and making a difference, small biotech and pharmaceutical companies can be a really interesting place to go. Next slide. Okay, and this is, I've worked at 3 pharmaceutical companies and started a medical device company and had a variety of other things. What I would say is none of them have ever been in the same sort of therapeutic area. So this little company out of Roswell, Georgia, we have had products for organ transplant infections. We've had a product that prevents fetal rejection. We've had spasticity products for studying cancer, rare diseases. And so what I think is one of the things that maybe is a misconception in our industry is that people believe you need to, be an oncologist to be in a company that studies cancer medicines, or you need to have a really in-depth background on some specific type of disease state. And what I think you actually have to be really good at instead is, do you understand basic biology and how it works? Do you have the ability to use data to sort of solve problems, map processes, find ways to improve? And do you have the interest in reading things that get you up to speed? So, in my job, I probably spend, I would say every week I read at least 10 journal articles that have to do with diseases that I don't know a lot about, but I'm trying to get the basics right. And then trying to understand what medicines might work there, or if we think something might work that's a competitor. And so I think to the extent people have a broad range of background in, biology and math and chemistry, like they can really get up to speed on a lot of things, and they don't have to go super deep right away. For me, that was important. I remember when I was a kid, and I talked to my parents before going to college, and I said, I think I might be a doctor. I'm really interested in this stuff. And they said, no, you need to get a job. And so I think, most people are probably on the side of things where it's like, okay, well, I'm going to be practical. I'll go get a degree and then I'll go to work. And so, for me, I think this industry is pretty interesting. There are a lot of places to go, but it relies on some common skill sets that we'll touch on today. Next slide. This is what is SAIL now. So like I said, we have 4 clinical programs. All of those studies read out between now and January. So it's a pretty exciting time at our company. We read out of everything in January, and then we've got to go raise money. Maybe we'll try and go public, or maybe we'll, be able to raise money through private markets. But that's kind of the biotech thing. You sort of run a study, you see how it goes. If it goes well, you raise capital, you run the next study, and you try and eventually get to where you're marketing products again. And so right now we're studying spasticity, typically associated with like traumatic brain injury or spinal cord injury. Knee osteoarthritis, which is a very large market. A lot of people have this problem. We think roughly 30 million people in the United States do. And then a very rare disorder that affects about 2,000 individuals in the United States, and then a rare brain cancer. So, we're hoping to have success over the next year. And as you can see, we have a really broad ranges of things we're studying. We only employ one physician at our company. And so, the team has been able to kind of learn and, find ways to sort of understand what science is appropriately applicable here and apply, how the business side might be along with the science in order to get something through the clinical studies and potentially the market. Next slide. One of the things they said is, can you cover something about what jobs might look like? And so, in our industry, in Georgia, if you are someone who has no prior experience, your starting pay, if you come out of college with a bachelor's, typically in a scientific field, and you go into sort of a research and development or marketing and potentially sales organization, I would say in Georgia, you'll make probably $80,000 or more if you find the right type of role for yourself. It's a lot more in Boston and San Francisco. The cost of living there is also a lot higher. But the big hubs where, they attract talent are Boston, San Francisco, and San Diego. Those are the largest sort of biotech hubs. North Carolina has a decent amount of jobs, and then we are trying to really build that out in sort of Atlanta, also in Athens, where we have access to universities and a lot of excellent talent. So we're trying to build more of sort of a biotech hub here in Georgia. As it relates to manufacturing quality with a technical degree, probably 55,000 or more in a larger organization. So these are the types of companies that would have a manufacturing facility. We outsource all of ours, but there are a lot of companies that build things here. And so the big roles that people would be looking for would be manufacturing. There are a lot of roles in quality control in our industry, and they would typically require a technical degree, or also a lot of people are hired directly out of the armed forces. So you end up with a lot of folks who have been in the armed forces that end up in these types of roles, and they're good roles. And one of the interesting things about biotech, because it's sort of a growth industry, and is higher risk than maybe some other industries are perceived to be. Most people who are employed by these companies get stock compensation, should stock perform, or should a company go public or something like that. The key skill sets we're looking for, scientific understanding. Like I said, can people read information and understand sort of the basic biological processes and basic scientific principles? Can you interpret and present data? So most decisions that we make in our industry are based on our interpretation of data presented to us. And a lot of times those data are presented poorly. And we make bad decisions because of it. And so, to the extent that when people are doing their lab reports and understanding charts and, how information is presented, or even in journal articles, I think this is a really key skill set. If people can get good at that, they can be great in any company. People who have an excitement about science and disease, our industry is built on failures with a few great successes. And so, I've been in the industry, I guess, about 18 years. And out of those 18 years, I've always had a job, always had a great job. It's been interesting. And I think I'm batting like 4 for 13 of, drugs that actually did well in their studies versus failed and made it, and those that made it. So, this is kind of a place, if you don't like the business and don't like science and don't like health, it's a hard place to be. But if you do, there's a lot of opportunity. You just have to be able to roll with the punches. And then I would say the last thing is basic engineering concepts. To the extent that people understand process flows and process flow mapping, literally everything can be improved by about 40% in our industry. Whether that's internal finance processes all the way down to sort of, how much protein a cell produces in a biologic product. All of these things come back to basic engineering principles, where if someone can actually think in a process fashion, they can actually make a huge difference. And we'll go over some examples of that. Next slide. Okay. I won't say much here, but these are kind of the key components of our company and most pharmaceutical companies. And
everything in green that's there, we would have, can you guys hear me?
Yes.
Hello?
Yeah, it just got a little better. Like it
Okay,
went to
sorry.
Bluetooth, maybe.
And everything in there that's green just is basically a common skill set that we could use people who understand technical things and technical people with. And so here, I think something weird happened. Hello? Is that better?
Yeah.
Is that better? Okay,
Yes.
Great. Okay, sorry. Anyway, essentially every job in our company, with the exception of finance, in raising money, requires like a scientific skill set, or a scientific skill set is valued over other skill sets. Like we would prefer to hire someone who has an interest in science than someone who just has like a marketing degree. And we'll talk a little bit more about that. But I think, again, this is a place where you could be in sales and marketing and have a scientific background, or business development, or you could be, actually doing bench level science. Next slide. Okay, so I brought in two examples here that I hope to kind of bring some, I think people think of our industry as like there's like an epiphany, and then there's a big clinical trial, and then it's just like money. And actually, the vast majority of the industry is really complicated, highly focused on sort of quality, and really hard to find the right patients for. So there's a product that we bought from a company, CSL Behring, and it is Cytogam. It's an immune globulin. So if you go and gave plasma and you had a high rate of cytomegalovirus antibodies, then we would take your plasma and we would make it into a medicine, and then we would sell that to transplant centers to give to patients after transplant that have a donor. Maybe the person who donated the liver had cytomegalovirus at some time in their life, and the person receiving it did not. And so you would want to protect that person by giving them these antibodies so they didn't actually reject the organ. So it's a pretty important drug, relatively small and niche, but saves a lot of people's lives. And so what happened was the company who owned it was needed to sell it because they needed the manufacturing space. And so in order to transfer a product like this to a new manufacturing facility, you have to, quote unquote, find something that's like for like. So the FDA would say, we know how it was made in Switzerland. Wherever you're going to make it now, like we want you to make it basically the exact same way at every step, potentially even down to the same equipment. And so we traveled all around trying to find what facilities could do that so that we could make a like for like process. Then we engaged in a 2.5 to 3 year technology transfer process where we moved it from Switzerland to the new facilities. And the Swiss facility was also shutting it down and moving the line. So we made a whole bunch of inventory and then stopped producing it, and basically had this gap where we had to do the process transfer and then get the FDA approval of that process. And so a little bit about Cytogam. I told you it was underutilized for a long time. And one of the things that we wanted to do was grow the product. There were a lot of people that should have it that were having, losing their liver after a transplant. that probably wouldn't have with this product. And so we had to like update the marketing strategy and medical communication and how we talk to doctors. And we were looking also at studying it for a new indication of preventing infections in first and second trimester neonatal or babies that were still in utero. And if babies that are still in utero, if the mother gets an infection during the first or second trimester, it's the highest rate of newborn hearing loss and a variety of these other things. So if you see children with cochlear implants, most of them their mother had cytomegalovirus in the first or second trimester of pregnancy. And so this medicine potentially given to the mothers would prevent that. So what we ended up doing was we bought the product, we moved it half of the process to a facility in Belgium, half of the process to a facility in Israel. And it took about 2.5, 3 years to do. And then we sort of revamped the entire marketing strategy and things like that. And so the product actually went from decline to growth. The NIH had initiated financing for this study in babies. And we were able to sort of manufacture in a new facility and actually keep the drug on the market over the long run. And so what that required was, a lot of work from a lot of team members. But in the next slide, I kind of talk about the key things that were takeaways here and how it might relate to the work that you guys are doing and trying to help kids develop skills with. So next slide, Megan. First was, ability to understand the disease and how medicine works. So we were trying to figure out if this was an important disease before we bought it. And so people were coming up to speed and trying to decide, is this declining because there's no need for this medicine anymore? Or is it declining because it's being poorly marketed and there are a bunch of people who actually should get this medicine? And if you don't understand the disease or how it works, you can't figure that out. The second was we had to map the manufacturing process. And so literally the number one thing that was required this manufacturing transition. And so process mapping, understanding of process flows, in my view, 80% of every job in my company, people would do a lot better at their role if they had this one skill set. So I would encourage you to kind of think this through and people understanding how, work items work and processes work. One of the things the FDA asked us to do was change all the lab processes. So because we were adding slightly different machinery, we were breaking the process up in different places. They wanted to make sure that the product was the same where it ended the process as where it started the next process step. And so we had to come up with all these different, lab processes on how do we test this for quality. And so, people doing work like running gels, you start to get a sense of, how all this stuff works. We aren't running gels per se in this process, but I think basic lab understanding and how these things work really helps a lot. Technical writing, everything we do at the FDA is basically done by a technical or medical, someone who has a skill set in that. It may they not be a writer per se, but they have done a lot of it. So, anyone who does lab reports and stuff like that, I think that's a really important skill set. And then, interestingly, people may not realize this, but the best salespeople are typically bachelor's degree science folks who like selling. And it's because they're talking to doctors. I mean, these are people who are out there talking to like world-renowned transplant physicians that are doing transplants in the hardest to treat patients. And convincing them to use a medicine that they think is old is not that easy. And so, you've got to learn to speak technically. You've got to be excited about what you're doing. And you've got to really study the science, because you don't just go and say, hey, do you have any challenging transplant patients? You should use Cytogam. That never, ever works, right? You actually have to talk to the physician about, tell me about your patients that have recently had a liver rejection. Okay, what happened? Tell me about, and actually work with the physician to understand what this looks like. And so all of these skill sets, I think are developed early on in sort of the science basis, and to the extent people can be excited about it now, they'll do well going forward. I'll go through the last example and then we'll stop for questions. And I hope this is helpful. Example two, I used to work at a company called Genentech, and Avastin was one of the medicines we had, and it's one of the best selling cancer drugs ever invented. How it works is it's an anti-vascular endothelial growth factor, VEGF. And what it does is it basically chokes cancer by preventing it from developing new blood vessels. And so it tries to grow, but it needs food, and it gets food through blood vessels. But if it can't grow the blood vessels, then it can't grow. And that's essentially how it works. And how it's made is you take genes that we've edited to make the specific type of antibody, which is Avastin. And we implant that into Chinese hamster ovary cells. We put them in a big, like 4,000 liter vessel, and we feed them, and then we program them to secrete this antibody. And then when that's done, you sort of filter out all the other stuff, all the cells go away, and you keep just the antibody. And anyway, what happened here was this was a highly successful medicine that had a whole bunch of new indications coming at it. And so we had to decide what we were going to do because we didn't have enough manufacturing capacity in order to manufacture this product and fulfill the demand. And so, essentially you could bring on a larger factory, but that takes 3 to 4 years and $300 million. You could try and improve the process. No one knew how much they could improve the process because this was one of the first really high volume antibody products. And so, I happened to be at Genentech at the time. I had just joined. And so kind of what happened? Well, next slide. This is where things are a little different than Sale. They did both. We don't have the opportunity to do that. But Genentech paid Lonza to build a facility in Singapore for them for $290 million. Separately, we initiated a process improvement campaign. And the results of the process improvement campaign were incredible. I think for people who may have worked in the auto industry or something, they'd say, oh yeah, of course that would have occurred if you didn't put the Toyota production system into it. But I think in our industry, that's still relatively new on manufacturing improvements. So we had about a 4x improvement in efficiency of getting the DNA into the cell. So we had viable Chinese hamster ovary cells versus dead ones or ones where the DNA didn't go in. And number two is we doubled the efficiency of the chromatography techniques. So chromatography is basically the filtration system that we use to get the antibody out of the rest of the vat. And so we were able to, quadruple the efficiency of producing antibody, double the efficiency of removing the antibody. And then we cut the total time it takes to make a batch by 65%. So in the current factories that we had, we could triple the number of batches that you'd make a year. And this was done primarily through a few key efficiencies. The first was we designed better feeding techniques. So we gave them slightly altered. We did a bunch of experiments on, how much sugar, what types of sugars you give when you feed the cells. And then we also altered the shape and speed of the paddles in the bioreactors to mix it. Because what happened was we were mixing too quickly, creating too much shear force and killing a bunch of cells. And so we found a way to kind of more slowly mix these, but mix them more efficiently. And so they were fed better. And we also had fewer cells burst. And then a lot of it was actually reduced downtime between cleaning and maintenance. So in 18 months, all that was done for about $12 million as context. Okay. And why I have such an interest in sort of this process mapping. The plant in Singapore was qualified, but they actually never needed it for Avastin. And they've never used it for any Genentech products. They actually only sell other products. They qualified it and then manufacture products for other companies. And so kind of what's the takeaway here on the next slide is process mapping. That was like a huge focus of this improvement. Understanding how it works. And this is where simple things like cell mixing and feeding, people can design those experiments and understand it. Optimizing chromatography steps for the protein, all that stuff worked. When they actually went and found out what the process is for cleaning a bioreactor and the downtime, they found out that they didn't have enough hoses in the facility. So they spent like $8,000 in hoses, and they were able to double the capacity of a $300 million plant just by doing that. And so, between that design of experiments, process monitoring, they did a lot. And all of that was far more than you'd get out of sort of, the finance team said, hey, let's build a $290 million facility. And so I think the key takeaway I'd like to share is sort of, you go to the closing slide here, is there are so many things in this industry that can use folks who have, in-depth PhDs, or they have a technical degree, or they just have an interest in science. One of the members of our regulatory team has been in the industry a long time, has her high school diploma, and she's our director of regulatory at SAIL. So, I think this really comes down to the work that you guys are doing is awesome work. And I hope that you can see that the things that you're working on, technical writing, process mapping, interest in science, those are the things that drive what we need in our industry and from our employees. And happy to answer any questions that you have.
Yeah, so you guys can shout out if you have questions.
Hey, this is Pamela Consuegra. For technical writing, for my biotech classes, we just use the Bio-Rad textbook as kind of a guide, and I don't have any industry experience. So can you kind of summarize what technical writing means in your space?
Oh, Dave, did you mute?
I didn't mean to. Can you hear me now?
Yes, we can hear you now.
My video stopped. I don't know why. Okay, I'm having technical issues. I apologize. For me, it's not like, can you write a journal article that's going to be published? It's really like a lab report, is the way I think of it. Like, if you can technically describe things. So the vast majority of our technical writing is what goes to the FDA. And what goes to the FDA is typically, here's some background on the disease of the process. Here's what we're trying to achieve. And then here are the process steps in detail that we've taken to do it. So I think of it very much like if you were doing a science lab report where, let's say you're in a physics class and you're rolling cars down a ramp and you're raising the level of the ramp to see, the speed of the car or something like that. And going through the process of describing those things and putting data in data tables, that's really what I think of as technical writing.
Thank you.
Mm-hmm.
Any other questions for Dave? Well, he shared his email, so I'll put that in the links for today too. So if you guys think of any questions later, you can send them Dave's way. So thank you so much, Dave, for sharing with us. That was really informative, and I'm not surprised that they don't have many questions because you were really thorough.
Oh, well, sorry, probably too much though. Well, anyway, I'm excited about what we do. And I'm really thankful that you guys are educating all these kids out there. It's a great way to make a difference, I think, in our industry, and would love to have more people that are interested in science.
Awesome. Thanks, Dave.
Thank you. Take care. Have a great day. Enjoy your electrophoresis.
Thanks.
Bye.
Bye. All right. And we are going to stay in this room today, but we'll be turning it over to Mark and Trisha. And they're going to walk you guys through setting up your gels. And then once your gels are set up around 5:20, we have another speaker coming, Lori Kreimeier from Boehringer Ingelheim Animal Health. So I will let Mark go ahead and get started. You can unmute. There you go.
Hey, everyone.
Hello.
So if you could turn to page 22 in your binder for the STEM electrophoresis kit. And it says student manual and background. Mm-hmm. And then we need quite a few items. You're going to need your gel from last week, bag of Skittles,
Medicine cups.
your 4 medicine cups, 2 paper clips, Your reference dyes, they're in the small microcentrifuge tubes here. Let's see.
Transfer pipette.
Transfer pipette. Your dye extraction solution. And then you should have 5 9-volt batteries.
And this thing.
And then your wires. So Megan, we have about 15 minutes, right, to get this set up?
Yeah.
Okay.
No big deal. We can have Lori wait a few if needed.
Okay. So please let me know if I'm going too fast. But the first thing we're going to do is take our pack of Skittles here. Grab 4 different colors.
I'm trying to get, yeah,
there you go. And place them into the medicine cups. Then get your transfer pipette. And where Trisha's pointing there, that first increment on your pipette is 0.5 milliliter. And that's how much of the dye extraction solution we want to add to each medicine cup. And you're going to put it directly on top of the Skittle.
I'm sorry, can you repeat one more time? Last
Yes.
step?
Yes. So you take your disposable pipette. Good. No, actually this one here. Let me see.
The plastic bulb one.
Yes. And that very first increment you see is 0.5 milliliter. And that's how much of the dye extraction solution we want to add to each medicine cup directly on top of the Skittle.
Okay.
And then after you add it, if you kind of shake it around a little bit, it helps get more of the color off of the Skittle.
Which one is the dye extraction solution?
What's the solution that we're putting in?
Right here it's in a microcentrifuge tube.
Okay.
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 And basically the extraction solution is going to help load the gel. It's going to make it heavier so it falls down into those little wells in the gel.
Excuse me? And how much the solution do we add? I couldn't see where she was pointing.
It's 1/2 of a milliliter, which is that first increment on the transfer pipette.
Should say 0.5.
So how many drops do we drop in each medicine cup?
0.5 milliliter.
0.5 milliliter in each thing?
Yes, in each medicine cup, right on top of the Skittle.
Thank you.
You're welcome. You want to get the electrode set up before pipetting.
Yeah. And we need to pour the tea.
And then you should also have your 1X TAE buffer that we made last week. And the purpose of that buffer is to help conduct electricity. So as Megan said at the beginning of this session, with the simulation, we're going to use electricity, and we're going to use our agarose gel. And she had mentioned it's like a molecular sponge with pores. And our different dyes are going to travel different distances based on their size. So the smallest ones will travel the furthest through the agarose, and the larger ones will not travel as far. And it's also based on their charge. So all of these have negative charges, just like DNA does.
And if everybody has their dyes ready, we can actually set up the chamber really quickly. And that's going to require you to grab your 2 paper clips. And you're going to need to kind of fold them into like a U. I have one end longer than the other. But you're going to need both paper clips.
I'm sorry, but what do we do next after adding the dye-X solution?
Just take each medicine cup and just swirl it around a little bit.
Okay.
And then we're going to get the paper clips. I think if we flip the page. As far as the paper clips, which will be our electrodes, page 28 has a diagram. Yeah, I'm looking. Summer. Is everyone good? Again, I just don't want to go too fast.
The paperclip is proving more challenging for me, but I've got one down. I'm like, I need a hammer. Bend it into a U shape.
Can you show us under the picture what they look like again?
I have one of mine that's one end that's a little bit longer. I find it's a little easier to put the little alligator clamps on, but a U-shaped with even sides is fine. I don't know if you can see mine, the background there.
Okay, now the next step, and when you're going back through and looking at this later, there's a step where it tells you to cut part of the gel, but what we've been doing is simply pushing the paper clip down into the gel.
Just on the end.
Yep, on the very end. If you push it down, just push it right in through the agarose. And then at the other end, do the same thing.
Is it okay if it doesn't span the full width?
Yes.
Okay.
I stored my gel with extra TAE buffer on top. Can that just go down the drain, the extra buffer that was stored on with it?
Yeah, you can put it down the drain, or we're actually about to pour buffer on top of it anyways. So you can just leave it too.
That was
I think what's happening is we're not able to see the demo a lot if somebody has their microphone unmuted, then it switches to their camera, I think. Or is there a way I can like freeze it on the, I can't figure out the Zoom.
If you click on the three dots over them, you can say pin speaker or pin them, like click on their three dots. I tried doing that on my end and I thought it would change it on your end, but maybe it didn't.
Oh, let's see. I guess I can't, wait. On this app, I'm on my tablet because the camera on my Chromebook does not work, or my schoolwork, so I'll wait till they pop back up.
If the
And
pins are actually sticking out, is that okay?
Yes, that looks good. And page 29 has a nice schematic diagram showing the paper clips on either end of the box.
Does one side of the paper clip need to be all the way in the gel, or does it matter if it's sticking up on both sides?
No, it can stick up. You want it to stick out of the box because that's how we're going to hook in the alligator clamps.
Right, on one side, but is it okay if it sticks out on both sides?
Yes.
Like the other half of the U?
Yes, that's okay.
Okay, thank you.
You're welcome. Okay, so Trisha's going to take the 1X TAE buffer we made last week, and she's currently pouring it over the gel, ensuring that the buffer goes into the wells. And you just need maybe a centimeter or so. It just needs to cover the entire gel. So now this gel has the TAE buffer over it.
And you said it just needs to cover, it shouldn't go above the gel.
It should fully cover the gel and also go into the wells.
Okay.
A thin layer.
Yeah, just a thin layer over the top of the gel. You want to do that little drawing?
Yeah.
Okay, so now we're going to use our pipetting skills that we practiced in the very first session. Okay, so if you would find your micro pipette and please set it to 10 microliters. So as you're reading the pipette, it'll read 100. And then Trisha's going to show you something here. She's going to do a little diagram. This is very important.
I'm trying to work upside down like Da Vinci and backwards here on this camera. But basically what I've tried to draw is pretend that this is the actual gel, agarose gel in the plastic soap dish. And then this represents the wells that you made with that gray comb last week. And so it's a little bit challenging because the tip of the pipette needs to be in the well, but not through the well. So you might want to take a minute and put a tip on your pipette and then just kind of feel and get an angle for what it looks like, where you can see that the tip is in the well, but it's not like poking through the well. You're not stabbing through it, but it's in the well. Because if it's above the well in the TAE buffer, it's just going to spread out all around. And if you poke through, you're going to poke all the way through the gel, and the samples aren't going to run properly. Does the diagram make sense, and kind of getting used to where the tip is in the gel? And I could never be a neurosurgeon or anything. So when I do it, I usually have to like brace my elbow so I can get, so I'm not like too wobbly.
Yeah, if you do this with the students, they want to do what they see on TV. And what we do is brace that arm. And then it's a good time also to get a feel for the different stops of the pipette. So you want to go to the first stop, put the pipette tip into the reference die, and I'm going to show you this. I'm just explaining it first. So I push down to the first stop. Then I go into the reference die, and that's one of those 4 tubes that you got out. Those are your reference dies. We're going to put all 4 of those into the wells of the gel, followed by the 4 skittles. First stop, insert, and then slowly let off the plunger, slowly on the micropipette, drawing in, again, slowly, the 10 microliters. If you do that quickly, you're going to end up with bubbles, or it's going to be up higher in the tip, so you're going to have an air pocket. And you don't really want either of those. So slowly let up. I'm going to go ahead and, you want to do that?
Yeah.
I wanted to do it.
I want to do it.
And then I always visually inspect the tip. So you can see Trisha has the 10 microliters. And then what she's going to do is she's going to put the tip into the well, just like her little diagram. And she's going to slowly expel the fluid into the well. And here's a common mistake if you haven't done electrophoresis is once you expel all of the liquid into the gel, keep your thumb down, keep it depressed, and then slowly pull it back out. A lot of students let the plunger back up, and they end up basically sucking back in everything they just pipetted. Another thing that happens often is pushing down all the way through that second stop and doing it too hard. You're going to force all of your solution that you just put into the well. It's going to spill out into the buffer.
So my red doesn't have anything in it.
Okay.
So mine's
almost the same too.
My red is horrible.
Yeah, tap it down so you can take the tube and bang it down on the counter a couple times. Because there were 15 microliters in there, so you should be able to get 10 out.
It's also like if you're running out of ketchup.
All of mine. I put it in the centrifuge real quick, and like the red had like none left. It was
Nothing.
I don't know what happened to the red.
Okay, I had QA on these, so they should have had stuff in them. But maybe it evaporated.
Yeah, on
mine
occasion,
barely had enough.
and on occasion, those
So what should
topsoils,
we do?
those topsoils sometimes pop open too.
So after
Yeah, I
you
have the same issue with the red. And also, are we supposed to put the colors in like a particular order like y'all are doing, or does it matter the order? the order doesn't matter, and once you feel comfortable with one, then you can do the rest of the dyes, the control dyes, and then you can start using your Skittle dyes, and you can load those.
Thank you.
Now we change the tip for every time we do it, right?
Yes,
Yes.
changing the tip is ideal. If you have a huge class, sometimes letting them practice, we don't really stress to change the tip, but changing the tip is ideal.
Okay. My apologies. And I know we already went over pipetting, but it worked for me last time in that lesson. But when I pipette, it's just popping off the bottom part and it won't let me. Am I doing
Okay.
I'm doing something wrong?
Yeah,
We didn't do that last time.
make sure you're using the big. Oops.
Look,
Sorry.
oh yeah, that part. Yeah, the big one. That's what happened. Yeah.
Okay. Yeah, this other one is for ejecting the tip.
Yeah. Thank you.
You're welcome. When can I do some?
You can't. I feel like you're judging me.
So we're going to do 8 samples total.
Question, please. In regards to the colors that we receive, how much should we actually receive? That is these.
15 microliters.
The
And we're
question
going
is,
to we're going to add 10.
sorry, I have blue, yellow, and I see one seems to be red, and I have another one which is empty.
That one should have been orange or yellow 6.
Oh, okay. All right. Thank you.
Okay. Well, if you don't have it, no worries. You'll still be able to compare to the other ones that you do have.
Okay.
Compare those against the Skittles. Sorry about that.
I have a question. What type of dyes are these that are the standards that you're using? Can you just use food dye, or does it need to be something specific?
So they are food dyes. So like red 40 is the red 40 that food dye that you would find in food. I think they're weighted a little bit, so you could just mix in sugar water. I know Trisha, you said you did a bunch of different trials for well, it was for the dye extraction, but same thing.
Yeah, and a little sugar water was enough to weigh the dyes down. And if you teach a chemistry class, and if you actually look up the different colors, like yellow number 5, or what are they? Yellow number 1, blue number 5. You can find the chemical structure and the charge and everything, and then compare them. It's pretty cool. And I think
Yeah, it's in the
book.
binder. It's one of the
Okay.
earlier pages.
Well, you want to start these?
I do, but I don't think everyone's ready quite yet. If you are close to being done, you can get your 5 9-volt batteries ready. And while you're pipetting, I'll tell you, like Megan said, you can use sugar water. You can use other types of candies, like hot tamales and just various types of candies. And you can do this with just sugar water. you can get the different food colorings and again, add sugar water.
I don't, just food coloring.
Yeah, just straight.
Yeah, just straight food coloring can work too.
The TAE buffer, I've used tap water with baking soda dissolved in it. And it works fine.
Okay, I was able to get the green little thing like that with the green Skittle. I mean, not green, the orange, and then do it again with the yellow. Would that be enough for me to at least see some results?
Yes.
Okay.
Okay, so on page 29, there's a diagram for how to set up the 9-volt batteries. So we're going to have a 45-volt system. And these batteries, they can be used over and over again, as long as everything is disconnected. So Trisha's getting her battery array set up here.
You just kind of make a pyramid.
It looks like there's still quite a few pipetting. Yes. I see some.
Yeah.
Okay, so what Trish has done is she's taken her wire and she's attached the black alligator clamp to the negative side of the battery and the red to the positive. See if we can... So hopefully you can make that out. And again, the diagram in the book, it clearly shows how to set them up. So our dies that we're using have different sizes. They are different sizes, but they are all negatively charged. So it is very important that we hook up the negative electrode to the top of the box where the wells are.
And you might need to rip your cord apart a little to make everything easier.
So we're going to hook the negative side to the top of the well. And there will be a repulsive force that forces the negatively charged dies away and out of their wells. Now, if you hook it up backwards, they'll actually go the opposite direction.
And remember, we're doing chromatography, so we're separating these molecules out. So if you have something that's a secondary color, like with food coloring, you should end up seeing the two colors that make up the secondary color. What sugar solution concentration? I don't think it doesn't take much sugar.
It's very forgiving. Everything Trisha tried, she had Tris buffer, EDTA buffers, all these different PBS, and all of them worked.
And so we should start seeing like little bubbles, right, around the U part of the.
Yes, you should see bubbles. And when you see that, you know it's working. Double check to make sure you have the the negative hooked up at the top of the box, closer to the dies, and the positive at the bottom of the box. And within a few minutes, you should see the dyes begin to migrate through the gel matrix.
Yeah, you can already see yours.
And last year, somebody had asked what happens if a student puts their finger in the TAE while it's running. And the answer is nothing, because I did it. I had to try. So it's very anticlimactic, nothing, you don't get shocked. So.
And you will notice the paper clip on the positive side beginning to change color. And that's a reaction with the galvanized metal. And it's actually an oxidation reduction reaction for those of you that teach chemistry, electrochemistry. And it'll continue to change color over the next 20 minutes or so.
You didn't laugh at the fact that I put my finger in there.
I did, but I was muted. And I was going to say, I'm surprised it was you.
I was talking about Bart. He didn't
not the
laugh
spouse.
Bart didn't laugh at me. Somebody has to take one for the team.
Does anyone have any questions? Because at this point, we're going to let the gel run for 20 minutes or so.
Can we throw away all the little tiny containers and our Skittles and all that? Are we done with that?
Yes, we're all done with those. And the pipette tips.
You can rinse the medicine cups if you want to keep them. All right. So you guys can leave them running for 20 minutes. If you have questions, feel free to direct message Mark and Trisha or just pop them in the chat. So we're going to have a speaker here in a second. And you can leave your gel running until you're satisfied with how far it goes, and then just unplug one side. That will break the circuit. And then you can snap a picture of it. And we've noticed it loads better on black, and then you'll get a better picture if you put it on a white background. So again, questions can go in the chat. And now I would like to introduce Lori Kreimeier from Boehringer Ingelheim Animal Health, and she's going to tell you a little bit about BI.
Hi, everybody. Can you hear me?
Yes.
Excellent. I have to say, I'm really glad that you guys were running a little bit behind, because as I listened to how to set up these gels, it just reminded me of training back when I was in college. So thank you, number one, for that. And number two, how scientists should be trained. I wish they had these level of commentary, these level of detail as new students were coming into our field, because this is phenomenal. So I have to thank you for letting me be a fly on the wall for the last few minutes. Good luck on your gels. I know they're going to be fantastic. So I'll jump in because I know we only have about 20 minutes, right, Megan? We'll try to do this quick, if that much time.
Yeah.
All right. Let me share my screen for a moment. Let me know when you can see my screen. Should see a slide presentation in the background.
Yes.
Okay, let me put this in full mode. So I just wanted to give you guys, well, that's not it. So I'm going to back out of that. I never know how to do this in Zoom. I'm a Teams person. Teach this to your students to know how to flip between different platforms. So I'm just going to talk a little bit about Boehringer Ingelheim and kind of drill down to what we do in the US, and then maybe share a little bit about how some of the techniques, some of the methods that you're teaching your students are used in our world. So I'll go briefly. Megan asked me to talk a little bit about the company and then what I do. And what I do is I'm the director of biological development for the US. So these are the teams that are developing vaccines and biotherapeutics for the animal health sector. So first off, a little bit about BI. You guys can see my screen correctly, right? May not be in full screen mode, but that'll help me flip through this quickly.
Yes.
BI was founded in 1885, so we've been around for a while. You might not have heard Boehringer Ingelheim, and that's the way you say it, is Boehringer Ingelheim. There's a lot of different pronunciations. Boehringer for the Boehringer family, Ingelheim for where we're based in Germany. So those are the two combinations of where the name comes from. So we're a research and development company. We commercially produce medicines for not only people, but also for animals. And then next, where are we? It's a global company. So although it's founded in Ingelheim, Germany, we're all over the world. In my area of R&D, there's about 10,000 scientists, leaders, employees across the globe that do R&D, both for the human pharma and for the animal health world. It's about 53,000 employees worldwide. And then where are we? These are the major areas that we're in. These are some of the things that we do in the different areas. It's not only global research, we're opening new sites throughout the globe, both in human and animal health. We're known as experts in our area. So that's the other thing that's really important here, is that we not only are in this for industry to develop new products, but also to dive into the details, also to take that expertise and reach out and give back. That's another very important thing about Boehringer Ingelheim. So we develop small molecules and biotherapeutics, biologicals. You'll hear me say those words a number of different times, and I'll explain why in a few minutes. When I speak about give back, this is where our expertise comes into play. Research Beyond Borders is particularly out of human medicine, where we reach into different areas that are in need, not necessarily for profit, but because we have the technology to solve a particular unmet need in that area. So I'm going to jump through a little bit quickly about who we are. It's a family-owned business. And so Boehringer is the name of the family. And this is probably why you may not have necessarily heard about Boehringer Ingelheim. We are not traded publicly. You will not find us anywhere when you're looking for stocks. It's family-owned, and it will always be. The family is very interested in not only how we do things today, but how we will do things tomorrow and how it affects our planet. Innovation, you'll hear that throughout, not only from the way we develop new products, but the way we deliver those products out to the field, and powered by people. So I'll tell you a little bit more about people here in just a moment, where we are, what type of jobs there are within BI. I've got a slide for that as well. The other thing, being a family-owned company and wanting to make sure that that company that's handed down after generations to other members is developed correctly, whether it's for health for people and animals, whether it's for potential, how do we make decisions today that will impact tomorrow? This has to do with not only our communities. Sustainability is a very big thing with Boehringer Ingelheim, even the way we develop and construct new buildings. So environmental and energy design is taken into account as we build our new buildings. And that also overflows into more green. I know we're on a time frame, so I'm going to flip through these, but if there's any questions, I'm happy to answer those or try to answer those. So I mentioned 53,000 people. Where are they? By the regions, this is where they are. And by the different functions, by production, what do I mean? It's manufacturing. It's the people working in the factory to commercially produce those products, package those products, and ship those products. Administration, what does it take to make a company like this go around? And a lot of the administration requires a technical background, so a good understanding of not only our regulatory agencies, but the business at hand. So business and technology go hand in hand together when I encompass administration. Marketing and sales, of course. What are the needs of the customers? I can tell you, last week I was in a meeting with a number of different marketing and sales folks. They're DVMs, they're master's degrees. These are people that came up through biochemistry engineers, and found their way into sales and marketing. So surprising that they're not all MBA. they're not all business administration majors, that they have come through into our area because not only do they care about animals, but they know how to reach the veterinarians out there, and they like being face-to-face with our customers. Research and development, this is the area I'm in on the animal health side, and apprenticeships and internships. This is another really important thing for Boehringer Ingelheim. I'm going to jump to the next slide here, and this talks a little bit more about animal health. I'm going to drill down to the U.S. These are the different areas where we are in the U.S., so we're fairly spread out. Some of those areas are focused in a particular on a particular thing. Ames, Iowa, for instance, is a research hub for animal health. North Brunswick, they focus on small molecules. So if you have a dog and you use, I don't know, HeartGuard or Frontline or something, NexGard, something like that, that came from a small molecule or a North Brunswick research and development group. Athens, Georgia, that's where I'm based. Athens, we work on primarily pet vaccines and therapeutics here. Duluth, Georgia, this is our U.S. headquarters right there outside of Atlanta. Gainesville, we're very focused in the poultry area, poultry market, poultry commercial production and delivery of those vaccines for birds. Barceloneta, where we do a number of our chewables, small molecule chewables that are delivered orally. And St. Joseph, Missouri, which is one of our largest manufacturing facilities. St. Joseph, Missouri can produce not only for US and rest of world, but also for Europe. And so when you think about some of the lessons where you're talking about agencies, USDA, EMA, FDA, these are different agencies that might regulate the products that we produce. And the next one, this is a little bit more about the species. So we're across all species. And this is important because some of these markets have changed a lot. You may hear in the news about new emerging diseases that affect swine in one area in China. That can certainly affect your market and the type of vaccines that you use or the sales that you have in that particular area. If you're hearing about it as emerging disease, we usually have our interest peaked or are already working on it. Avian influenza would be another one that you might hear about. Foot and mouth disease, which thankfully we do not have in the U.S., would be another one that you might hear about there for cattle or swine. Pets is an important market. It's changed a lot. You think about that, think about COVID. We had more pet adoption during those COVID years. And that has really changed our business to a degree because pets very much they often get diseases that are very human-like, and those technologies overlap. Diabetes. I mean, there's so many different things in pain and inflammation, cancer that our animals are getting as well, and they're living longer and longer. And so these areas are really where we get a lot of synergy between our human health and our animal health areas. So then this just, a little proud moment here, I've got to say where we are as far as our competitors go. This is just an example of where we are for different types of products, whether it's parasiticides, think flea, tick here, therapeutics. These are the ones that affect diseases. So pain and inflammation, oncology, immunology, fibrosis. That's what we mean by therapeutics. Vaccines. If you got a cat, dog, horse, you are very used to vaccines, just like our kids. Fundamental core vaccines, and then always we're also looking at improving and developing new vaccines for the future. And then of course keeping our food source very safe and healthy. So our motto is around prevention, and that really is that's what really drives us, is preventing new diseases, not necessarily treating those diseases with antibiotics, for instance, but preventing the diseases from occurring. And then lastly, and I want to be conscientious of time, just some of the diversity here, and this is really why I love being in animal health. I mean, the type of products are so diverse, from a poultry vaccine where you have 1,000 doses in a plastic bottle that you're delivering in a poultry house, to whether it's in water or directly injected, to cancer therapeutics, melanoma. We have a DNA product for melanoma in dogs. I mentioned NexGard, which is one of our newest ones, and how we treat the parasiticides. So when I look at this table, what I think about is, yeah, way to go, cool, some brands we all recognize, but I also think about the technologies that is behind that, whether it's a bacteria or a virus that you grow and inactivate and purify and bottle and then deliver to the vet, or a viral vector, which may be live, and a live attenuated viral vector, where you're really just trying to amplify a particular gene product from that viral vector, to the DNA example that I gave you a few minutes ago, where you've got an E. coli with a plasmid inside that's pumping out, once you push particular factors during your production, pumping out DNA, which you then purify to deliver to an animal that's sick. So it's really kind of a broad spectrum, and that's part of the fun. I'm going to stop there for a moment with slides and start telling you a little bit about how some of these cool techniques that you're using are utilized in our world. Unless, Megan, do you want me to stop there for questions? Because I know we're at 5:50 Eastern, and we need to keep everybody on time with the gels.
Well, if you want to chat about that, maybe we can have people pop questions in the chat, and then we can get right to those.
However you want, and feel free to interrupt if you'd like to as well. So I mentioned I do biological development in Athens, Georgia. So I have a team that does upstream and downstream processing. So late stage product development, formulation of new products, and all sorts of those are the aspects of development. We also have a team that does analytical development. And when I think of things like Western blots, running the gel electrophoresis that you're running today, where you need to separate different components to analyze what's in that particular product. ELISAs are another big thing that we would develop in our analytical development. We also use tools like HPLC, mass spectrometry, and a lot of what's important is being able to not only know the technique, understand, realize that you've learned about it, understand what the output shows for a particular product, and then be able to sometimes develop those techniques and transfer them into a quality control environment where they're using that to release product. So that's when I think of analytical development, that's what I mean. Formulation development is it's taking the end product from upstream and downstream, whether it's a bioreactor process, whether it's the cells that you've grown to grow virus or bacteria that you've amplified, then the purification of that product. Now the formulation team takes that and they put that together in a safe and efficacious and potent manner into vials. And then that is what becomes in the future a vaccine or a therapeutic that's utilized. So these are just some of the ways that we would use those techniques in the lab. I mentioned also about regulatory agencies. So our field is heavily audited. USDA comes in, auditors come in, they all have a scientific background. We work with different agencies, like I said. And so I think that a lot of the tools and tips that you're learning today, and being able to do them yourselves, I mean, kudos. You're not teaching it from a book, you're demonstrating it with output. This is exactly the way teaching should be. And I say that as a daughter of a mother who taught high school science and chemistry. I mean, she taught for years. I remember growing up, going into the schools and getting to meet the other teachers, and I don't know, cleaning blackboards back in the days when we had blackboards in the past. And still seeing some of the students and some of the people in the community that she taught and which areas they've gone into. So any chance I have to talk and answer questions for teachers, count me in. Megan knows that. And I'll stop there for a minute because I know it's 5:53, and just let you guys ask some questions if you want to.
Yeah, well, and I'll start with one question. So you mentioned the internships and fellowships. What level of education do you guys look for that?
So it depends on the department. So I mentioned we have manufacturing, and we have in Athens, Georgia, and all the teachers for all here are based in Georgia, correct?
Somewhere in Georgia, yes.
Somewhere in Georgia, okay. Well, and we have areas all over. So our manufacturing facilities do not necessarily require the college degree. They may take apprenticeships, where people are able to come in post high school and be able to train. We also are working very closely with some of the groups that are developing certificate programs and high school curriculum, so that students come out with tangible techniques and can apply for jobs directly. So in our manufacturing and in our quality control departments, we would have opportunities for students to come in directly out of high school. In R&D, and so that could be high school or 2-year programs, so associate's degree. Many people in manufacturing also apply with bachelor's degrees, but that kind of gives you an idea of the diversity for applicants. In the R&D department, usually we're looking for people with graduate degrees, so at least bachelor's, but master's, PhDs. On the team here, we have, veterinarians that have gone back to school for graduate work. I did my undergrad at University of Georgia and a master's program at University of Georgia. And I've been in this field for now almost 30 years. So you can do a lot. And I should also add, I went back to school using the continuing education program at Boehringer Ingelheim. So I worked and went to school, and the company paid for it to get my master's degree. So that should give you a little bit of an idea of that.
Yeah, thank you. And then I know Lori because we've been working together in Athens at the Career Academy on starting a biomanufacturing pathway, which will be under manufacturing in the DOE standards in CTAE.
Yeah, absolutely. Sorry, I just got distracted with the chat. I just opened it up. So glad that there's some customers out there too. I know I am as well. Were there other questions, Megan, or did I miss a question that you asked me? The ACC career pathway is an important one for us, at least locally for Athens, and it's one that we'd like to see broadened as well. The other thing I think that we have very much enjoyed, and we had our charter year this past summer, and we will do this in the future, is have teacher externships in the summer. Because I think coming into a company and spending a week, and not a whole week, but a few days during that week, and touring through the different departments and seeing how what you're teaching applies to every day for the students allows you to kind of bring that into the classrooms, navigate some of your students towards areas that you think that they could excel in based on their interests.
Awesome. So how to apply for the teacher summer internship. So I guess, can you send me that information when it becomes available?
Yeah, absolutely. And Megan Wesley is our connection there.
Okay,
and
perfect.
This is something we'd like to broaden here in the future. So Wesley Molina is our connection there.
Yeah, and we can chat about the gift program that Georgia Tech offers. So it's a like 6 to 8 week summer internship for teachers. So yeah.
Absolutely. Awesome. So someone worked in the animal hospital for 5 years and at USDA. Ah, good. This is great. I mean, I can tell you from the auditors that we've met, they have all taken different routes to their career. I told you a little bit about mine. I've been with the company for almost 30 years now. The company's changed names 3 times. We were a small company, and then we were a joint venture between Merck, which you've heard of, and Sanofi, and then Merial, and then Boehringer Ingelheim. And throughout that time, it's been in different roles. So the important part to think of there as you're talking to your students is you can take lateral steps to learn something new and apply it, and it's going to make you even more it's going to make you a better candidate for your next job. So not only applying for jobs that are lateral, may not be a huge promotion or a fancy title, but it's something new that you're learning. And then when you apply for your next job, that I don't know, ability to bring all of those different experiences in may make you a better candidate for the next one. So I know for me, I started here as in production. I started for 2 years in manufacturing. I went back to school working on my master's degree, got infatuated with live viral vectors. I could talk all day, but that's not the topic today, so I won't. And worked on that for my grad school, worked with CDC, with an external company on a project that was of interest to Boehringer Ingelheim. And moved into the R&D department after that. So I was a scientist, and then all of a sudden I moved to the next level of science. Then I had some direct reports, and then I decided to move into a different area. So I've kind of dabbled in regulatory and clinical, mainly focused in the research and the development groups. And I love equipment, and I see Jeff Rapp was on the phone, so he may know that as well. So when it came to kind of honing in on a particular area, bioprocess development kind of got my attention there. So when you start talking about bioreactors, filtration, centrifugation, chromatography, any kind of formulation, whether it's adjuvants or lyophilization, I'm in. So it doesn't have to be a direct path. It can take a while, and it's always learning something new and taking on additional responsibilities in every step.
Awesome. Do you guys have any other questions for Lori? Awesome. Lori, would you mind sharing your email with them?
Sure.
Hopefully they don't all get sent to spam like I did.
It is, I have to say, it is the longest email in the history of the earth, okay? Because my name is hyphenated and we spell out the entire name, Beringer-Ingelheim. So I'll type it in the chat. Or Megan, I'll just send it to you. You can plug it in there. You've got it. You can just plug it in there.
Yeah, awesome. Will do. I'll share it out with everybody in their links.
Of course.
And
thank you guys again for letting me be a fly on the wall as you learn to set up your chromatography. I hope it goes well. It's almost 20 minutes, so you should have the results soon, huh?
Yeah. Looking forward to seeing some photos of them. Thanks, Lori.
And you're welcome. Thank you again, Megan, for the invitation.
Yeah. Thank you. Have a great rest of your evening.
Bye.
All right. So yeah, everybody's gels are a little scorched, a little run. Hopefully you grabbed a picture of it. So if it sits around too long, the dyes will start to kind of diffuse out. So make sure you take a photo of it to preserve your memory of this, because it will not stay around for you. So yeah, Mark and Trisha, if you want to come back on and do a little recap.
Okay, so hopefully everyone's gels have run. And you can see the yellow dye, which those are the smallest dyes, have traveled the furthest through the gel. And the blue dye, or both of the blue dyes, are closer to the top. So they're larger dyes. And this also is similar to what we saw with the chromatography as well, when we eluted those different pigments from the Kool-Aid. So does everyone's gels look similar to what I described, or what you can see here with our gel?
Yes.
Okay, and so there's a lot of cool stuff you can do with the using dyes. So Trisha and I have a couple different labs that we run. One is on birds and phylogenetic trees, and looking at the similarity between birds, and then using electrophoresis to simulate the differences on a molecular level. And again, all it is figuring out the right mixture of dyes to place together, and then they will resolve or separate according to those sizes. And another one is our environmental DNA lab, which uses, again, this exact setup, but different recipes of dyes to simulate the invasion of Asian carp into our Georgia waterways. And we would be thrilled to share those resources with anybody.
I think Megan has them. And if you don't, Megan, we can send them to you.
Yeah, I have the eDNA one. I don't know if I have the other one, but I'll definitely put it in the Google Drive.
Okay.
So I have a question. You know how you can do paper chromatography when you're teaching photosynthesis and looking at the different colors of like your chlorophyll and your xanthophyll? Can you do that with gel electrophoresis too somehow?
Oh, I don't see why not.
Yeah, sure. You can separate any pigments, you can separate. The question is going to be the charge of the pigments. So sometimes you set up a gel where the chambers or wells, excuse me, the wells are in the center of the gel. And that way you're, as we have it orientated here, your negative dyes would migrate down toward the cathode, and your positive dyes would actually go the opposite way toward the anode. So I don't see why not. Any other questions or concerns? All right. Awesome.
Can you go over the cleanup process?
Oh, yeah.
Yes. So all you need to do, make sure you separate your batteries back, and the paper clips can go in the trash. The buffer can be poured down the sink. And then you can just use your hand and pull out the gel.
I gave it a good shake over the trash can yesterday.
Yeah, you might be able to get it to fall out if you shake it over the trash can.
And if you have a light tray or any kind of white light, students like looking at it under the light, or I guess it would be over the light. And then they also like breaking it. So I don't know, they think it's very satisfying. So if you want to give them the chance to break it, it's kind of fun.
I highly recommend giving your gel a nice little stab with your paper clip. Just kind of see the
Yes.
texture. It was rather unexpected.
Yes. And then that's it. Just rinse everything out and you should be all set.
Yep. So you guys can keep the box and the leads and the batteries to run this again.
And we do have write-ups for the labs we mentioned, and we will share the phylogenetic tree bird lab with Megan so she can post it as well.
Yeah.
I love the bird one.
And if anybody else has a lab that they want to share that's related to any of these, feel free to forward it my way and I'll put it in the Google Drive for anybody else. We've gotten some like cool pipetting ones. You do not need to bring these supplies in January. So in January, we will be using the mini PCR gel electrophoresis boxes. So yeah, for January, you will need your pipetter and your binder. I'll give you an insert with the rest of the curriculum so that you can put it in the same binder. So yeah.
And I would have to say that is my favorite part of the training is when we get to do this face to face. It's very lab intensive and it's awesome.
And you guys have already gotten to know each other a little bit, so it's nice to meet in person. And I'll send the suggested list of hotels later this week. We have a couple to choose, but there are many around that Central Georgia Technical College location. And yes, you do pay for the hotels. So the stipend that you guys will get, you can use that toward your hotel, or maybe your school would be nice enough to cover it if you ask. I'm happy to provide anything like the schedule or curriculum so that your school knows what you're participating in. So if you need documentation, please reach out and I'm happy to provide whatever you need.
All right. Well, I don't know, Megan, if there's nothing else, we're
I
all
think
set and
we
looking
are
forward.
all wrapped up. So you guys are welcome to sign off if you're good. And I will definitely hang out for a little while. I'm sure Mark and Trisha will too if you need to stick around and ask a couple questions.
Have a good night.
Have a great night. See everyone in
Thank you.
January.
Yeah.
Thank y'all so much.
Thanks, everybody.
Thank you, guys.
I actually did the pet by numbers activity with my kids today, and it
Awesome.
was great. They loved it. They had such a good time. So thank you for providing.
Yeah, that's great to hear.
Thank you. Bye-bye.
Bye.
Bye.
Thank you.
You're welcome.
Have a good night.
Okay.
Thanks to you guys. And hopefully you didn't have to rush too quickly to get back from DOE.
No, we were back. We left after lunch. Traffic was starting to get a little bad, so I was glad we left when we did.
Awesome. But yeah.
But
Yay.
thank you again, Megan. And January will probably crazy be here before we know it.
Yeah, it's gonna be a whirlwind. And then like I'm signed up to go to Histia, which is like the week after our training. I'm doing GSTA. We have Industry Insights in February. So it's gonna be like back to back to back.
Is the Histia one that's down at Great Wolf Lodge?
Yeah.
So one day I'll be there.
Awesome. Okay. So we're gonna have a vendor booth for base. And then I've been hooking them up with people for session. So like Elizabeth Proctor is gonna do one on one of the HOSA biotech events. I think Janet's gonna do one on Frugal Science Academy. I don't know if I hooked them up with anybody else. I'm not.
Yeah, I think I'm just there the one day where they go over like industry recertification stuff. But
Gotcha.
I'm not part of like the Histia, but the teacher we were with yesterday, she'll be there.
Awesome.
Yeah.
Yeah. Well, and then we just went ahead. I booked a room and Charlie and Lucas are going to come and do Great Wolf things.
Oh, yeah. I just said I'll just drive down for the day.
Yeah, it's not a bad drive. But
especially not from here.
But the booths, there's like an in-booth reception the like Wednesday night, and then the booths are all day Thursday or most of the day. So I needed to be there overnight anyway.
Right.
And they had rooms available.
Yeah, we actually have not ever been to Great Wolf Lodge, so.
I haven't, but it looks like a hokey child thing, so I've gotten more down with that.
Yeah, I bet it's super fun.
And yeah, an indoor water park in January, he'll have a blast.
Yeah.
But awesome. I think I can